ઓકે, આ ઇમસ્ક્યુલેશન ને બેગિંગ વાળી ટેકનોલોજીની વાત જે આપણે કરી છે લાસ્ટ ટાઈમ, રાઈટ? સો ડુ યુ રિમેમ્બર ધેટ ઓર યુ હેવ ફરગોટન એવરીથિંગ? યુ રિમેમ્બર્ડ ઇટ? વોટ ઇઝ કોલ્ડ ઇમસ્ક્યુલેશન? કોણ કહેશે? ઇમસ્ક્યુલેશન કોને કહેવાય? ઓકે, ઈડર સેન્ટર કયો જોઈએ? ચાલો. યસ, જ્યારે બાયસેક્સ્યુઅલ ફ્લાવર હશે, in that case only, when you remove the anther, that process of conversion of bisexual flower into female flower is called emasculation, right? So you are removing the anther, right? Begging कौन कैसे? Okay, भुज center.
फीमेल प्लांट में एक पेपर बैग कवर कर दो, फिर बीचा पॉलिनेट ना करें.
Yes, so to avoid foreign pollination, undesired pollination, to stop them pollinating with the unwanted pollen grains, we are covering them with the paper bags, and that's what called the bagging technology. So Mendel have used these kind of primary techniques or principles while doing the different experimentation of hybridization. Fine, so these are the primary hybridization technology. Okay, while discussing or while observing the different types of experiment, he concluded with 7 different traits into the pea plant. He selected pea plant. Why he selected pea plant we have, I think, discussed into the last lecture. It is easily convertible, easily culturable, it is easily crossable, it is easily, many offspring can be generated within short duration, lifespan is short. So there are many advantages what they are getting from it, right? So 7 different characters with 2 different contrasting traits he observed, and that's the list of it. Stem height, say for example, tall or dwarf. Tall is dominant and dwarf is recessive trait, right? So 7 character with 2 contrasting trait he got. So these entire list have been earlier described. Okay, now today we are going to start with monohybrid cross experiment. That is called, the event is called inheritance of a single gene, right? આ પણ થઈ ગયું છે આખો એક્સપેરિમેન્ટ? ઓકે, okay, so let us conclude with monohybrid experiment. Okay, so we are about to derive now about conclusion of the monohybrid cross. Okay, before we get into the conclusion statement, we must discuss these 2 phenomena called back cross and test cross, right? Okay, hybridization have got certain limitations. Whensoever there is experiments of hybridization, it has got some limitations. Say for example, while doing hybridization, it is very rare possibility that desired traits may be found into the next generations, right? So sometimes what happens is that, say for example, in monohybrid cross, when we have crossed these 2 parent, and we have got these two gametes. When it has been fertilized, they have produced the offspring of F1 with this genotype. So it is a tall plant, it is dwarf plant which has produced all tall plants, right? Okay, now usually crossing is followed from top to bottom method. But sometime researcher uses, say for example, F1 generation as one parent and the other parent could be either one of the generation which is earlier than that of current generation. આ જે કરંટ જનરેશન છે એનાથી પહેલી કોઈ બી જનરેશનનો પેરેન્ટલ પ્લાન્ટ લઈ અને એની હારે ક્રોસિંગ કરાવવાની વાત છે. સો વેન ક્રોસિંગ ઇઝ ડન એટ ધી પ્રીવિયસ જનરેશન્સ પ્લાન્ટ or parental plant, then it is called back cross. So either dominant or recessive parent is used for crossing with F1 generation plant. That is called back because you are going back to select the other parent, right? That is why it is called back cross. So do remember this phenomena. So back cross, it is a cross between hybrid and any of the parent. It may be dominant parent, it may be recessive trait carrying parent. Now comes the test cross. Now comes the test cross. So what is meaning of test cross? Yesterday did we have gone through this experiment or not? Test cross experiment? No. All right. Test cross is the cross where there is F1 plant but you are crossing it only with recessive parent, not dominant parent, right? So when you cross it with the recessive parent, then it is called test cross. એટલે એ back cross નો જ એક type છે. It is type of back cross only. But back cross can be either with dominant or with recessive. Whereas test cross is followed only with recessive parent, not dominant parent. That's the difference between them. So crossing of organism with dominant phenotype, એટલે જે organism છે એ dominant phenotype નો છે, that is capital T small t, with recessive individual. Recessive individual એટલે એ કયો recessive individual હતો પેરેન્ટમાં? તો કે recessive parent હતો એ small t વાળો. So when you are crossing it with them, it is called test cross, right? Okay, test cross શું કામ કહેવાય આને? સૌથી પહેલી વસ્તુ. Okay, so let me describe that now to you. What is the actual meaning of test cross? Okay, previous part, last slide. Okay, so F2 generation. Mendel has got two types of plants. One type of plant is tall plant, one type of plant is dwarf plant. Right? Is it correct or not? So 75% was tall and approximately 25% used to be dwarf plant. Okay. Now Mendel has done self-crossing experiment with dwarf plant and with tall plant. So dwarf crossed by dwarf and tall cross by tall. Now, when he crossed dwarf with dwarf, F3, he found all dwarf plant. F4, all dwarf. F5, all dwarf. So there is nothing, none of the progeny have been produced which shows dominant character. So what he concluded is, when there is recessive expression, it is called pure line, right? It is called pure line or pure breed. This can be either way used, pure line or pure breed. Okay, doubt? Doubt? Okay. Here, during F3 or F4 or F5, they can be tall or dwarf. Tall or dwarf. Tall or dwarf. There is a mixture, right? What it means? આનો મતલબ શું થાય? What do you conclude out of it? Okay, Bhuj Center.
સર એમાં રિસેસિવ અને ડોમિનન્ટ છે બે રિસેસિવ
Okay.
અને બધા પછી એવું હશે.
Okay, what you know is, when you again look it into the monohybrid cross, say for example, in a parent, it was capital T showing the dominant trait, that is tall. In F1, when it used to be capital T small t, they are also showing the trait to be tall. Am I correct? Yes or no? So, whensoever there is a two tall individual, it may be the possibility that it is either homozygous or heterozygous dominant. Che dominant, it is showing dominant trait, but we don't know whether it is homozygous dominant or it is heterozygous dominant. We have no conclusion, we have no idea. Understood? Yes or no? So there is a possibility that either of the tall plant have got that recessive gene which is remaining unexpressed into this progeny, either here or here, and that is giving the expression later on into the next generations. Clear thai che? Yes or no? So there are two possibilities. When the expression is, I repeat, when the expression is dominant expression, it can be homozygous or heterozygous. There are two possibilities. Okay, now any of the individual, I am giving you the tall individual, and if you want to decide whether it is homozygous or heterozygous, you want to check it, you want to test it, અને જે તમે ક્રોસિંગ કરાવો to test it, it is called test cross. Understood? So again, if you remember the definition of the test cross, તો એમાં તમારે કોઈ એક ટોલ ઇન્ડિવિજ્યુઅલને લેવાનું. સે ફોર એક્ઝામ્પલ F1, રાઈટ? એમાં ટોલ ઇન્ડિવિજ્યુઅલ તો કેપિટલ T સ્મોલ t હોય, રાઈટ, કેપિટલ ટી સ્મોલ ટી હોય. રેડી, F1 નો. પણ હું જ્યારે પેરેન્ટલ જનરેશનની વાત કરું તો એમાં કેપિટલ ટી સ્મોલ ટી છે. હવે આઈ ટોલ છે અને આઈ ટોલ છે. You have no conclusion, you have no idea whether given plant is parental plant or F1 plant. Fine. So what I give you the challenge is, I am giving you the tall plant and now you are supposed to tell me whether it is either homozygous or heterozygous. So to decide it, I have one specific technique called test cross. Right? So test cross માં હોય શું? તો I need to take dominant individual and I need to cross it with recessive individual. ઓકે, અહીંયા આપણે P કેપિટલ ટી સ્મોલ ટી અને એમાંથી આ F1 જે બન્યું હતું એની વાત કરું તો કેપિટલ ટી સ્મોલ ટી. સો આ કેવા હતા? ટોલ. આ કેવા છે? ડ્વાર્ફ. આ કેવા છે? ટોલ. સો વિથ ધેટ રેફરન્સ આઈ હેવ હિયર ધી ટોલ પ્લાન્ટ. ડોમિનન્ટ ઇન્ડિવિજ્યુઅલ ટોલ છે, મને ખબર નથી આ ટોલ કે આ ટોલ. રાઈટ, એન્ડ વેન આઈ હેવ ટુ ક્રોસ ઇટ આઈ નીડ ટુ યુઝ ઓનલી ધીસ. આઈ હેવ નો અધર ઓપ્શન. ડ્વાર્ફી એક જ છે, રિસેસિવ પેરેન્ટ છે. રાઈટ, સો દેટ ઇઝ ડ્વાર્ફ, રિસેસિવ પેરેન્ટ. અંડરસ્ટુડ નાઉ? So I take recessive parent, back cross કરું છું આનો આની હારે. So that is what I say. Now test cross. જ્યારે બેક ક્રોસમાં હું રિસેસિવ પેરેન્ટનો યુઝ કરીશ, it is called test cross. Ready? Doubt? All right. The example what is given into your book is this one. Test cross used to find out the unknown genotype of a character. મેં ટોલનું ઉદાહરણ લીધું, ટોલ અને ડ્વાર્ફનું. અહીંયા જે એક્ઝામ્પલ તમને આપ્યું છે, વોટ્સો એવર એક્ઝામ્પલ ગિવન ઇઝ ધીસ વન. રાઈટ, કલર ઓફ ફ્લાવર. રાઈટ, કલર ઓફ ફ્લાવર હિયર is, which one it is? કયો કલર દેખાય છે ફ્લાવરનો? વાયોલેટ. રાઈટ, it is violet color flower. It is dominant trait. But you do not know કે એનો જીનોટાઇપ કયો છે આનો. So you need to follow back cross, test cross. Now you are supposed to cross it with homozygous recessive parent. I repeat, homozygous recessive parent ની સાથે તમારે એને ક્રોસ કરાવવાનો રહેશે. So either it has two possibilities. Look at this now. It has only two possibilities. ये किधर गया भाई पेन? मिल गया. Okay. It has two possibility. Either it is homozygous or it is heterozygous. Right? Either it is homozygous or it is heterozygous. There are only two possibilities. So capital W or capital W small w. That's the genotype which is given there. Understood now? Okay. Now this is homozygous recessive parent carrying small w genotype. So there are two types of gametes here and there are two types of gametes here. These both are dominant, these both are recessive. So here dominant, recessive, that is capital W, small w, same way capital W, small w, capital W, small w. When there is presence of either dominant character, dominant factor, it will express as dominant trait. So one capital W, one small w, બધાયમાં એવું છે, all have the same genotype. So all will present dominant trait, બધાયની dominant trait હશે. So 100%, what we are supposed to mark out is 100% progeny shows dominant trait. Right, 100% progeny is showing the dominant trait. Okay, let us think about the other possibility now. Say for example they are heterozygous. So it carry capital W, small w, here small w, small w. So capital W, small w, capital W, small w, two are violet. Now small w, white, small w, white. So these both are white. So if 50% of progeny only shows dominant trait, then you can conclude whether it is heterozygous progeny. Understood? If 100% progeny is dominant, then it used to be, or it may be the possibility that this individual used to be homozygous. If 50% progeny shows dominant character, then it is heterozygous to be. So that's how you can decide. You can arrange the test cross and you can decide whether given dominant individual is either homozygous dominant or heterozygous dominant. Understood? So Mendel conducted this test cross to determine the F2 generation genotype. Right? F2 generation na genotype je છે capital T, capital T, small t, capital T, small t and small t, small t. Right? What he was having, the idea is he got F2 generation into two format. Tall plant and dwarf plant. Am I very correct? 75% tall plant and 25% dwarf. Now tall is whether homozygous or heterozygous. એને decide by conducting this test cross કર્યું છે. Understood now? So these test cross have been formed and designated by Mendel to know whether whatsoever tall plant he got used to be either homozygous or heterozygous. So what he concluded that 25% used to be homozygous, 50% of total is heterozygous and 25% is dwarf. That is homozygous recessive. Right? 25, 25, 25, 25. All are 25. That is capital T, capital T, capital T, small t, capital T, small t, small t, small t, all 25. So these are same. So it is 50. So 25% capital T, homozygous, 50% heterozygous and 25%. That is the dwarf plant, that is homozygous recessive. So that's what the conclusion he made using this test cross. So test cross શું કામ ડિઝાઇન કર્યું તો એનું આ લોજિકલ એક્સપ્લેનેશન અહીંયા આપ્યું છે. To know this, to know whether the dominant individual is homozygous or heterozygous, we are considering the test cross, we are arranging the test cross. All right. Now comes the laws of dominance. The first law which have been concluded, right? The first law which is getting concluded after conduction of monohybrid cross, that is called law of dominance. Yes, Bhuj Center.
સર ટેસ્ટ પ્રો એક્સરસાઇઝ પ્રોસેસિંગ કરીએ છીએ એ પેરેન્ટલ અસમ્પ્શન હોય કે કોઈ પણ હાલે
કોઈ પણ હાલે, પણ એમાં જીનોટાઇપ તો when it is recessive, તો એ તો ઓમે homozygous recessive જ હશે ને. So look here into the definition. Here into the definition they have not mentioned it whether it is parental or non-parental, right? અત્યારની definition. What I was telling Mendel has used recessive parent. That's what I mentioned. Mendel have used recessive parent. Understood now? So here crossing of an organism with dominant phenotype to a recessive individual. Any recessive individual you can take. All right. So first law of dominance. We know this law. Mendel have said earlier that there is something which is getting forwarded from one generation to the other, and he gave name the factor to that something. Am I correct? આપણે આની પહેલાના લેક્ચરમાં વાત કરી હતી. There is something that is factor which is getting forwarded from one generation to the other. This is the reason why we have pair of factor, because our cells are having two set of chromosomes, two set of DNAs, two set of factors. One is from our maternal parent and second is from our paternal parent. Both are giving us one set of the genome. So we have two factors. For each gene we have two factors. One is from maternal family, one is from paternal family. So there are factors. Factors are always in pair. Factors are always in the pair. Right. So that pair can be homologous or it can be heterologous. Right? So it can be, the factor can be into the form of homologous or heterologous. Okay. One character is controlled by one gene. That's what we are supposed to get now. Okay. कोई एक character है that is controlled by one gene. Any one gene has two alleles, that is factors. Any of the gene has two alleles. That allele can be same or different. It may be same, that is similar, or different, that is dissimilar. If it is same, it is called homozygous. If it is different, it is called heterozygous. Homozygous can be dominant. Say for example capital T same છે homozygous, or it can be recessive, small t, right? Both same, both are same. Now why it is heterozygous? Because in the pair one allele is dominant and the other is recessive, that is small t. So it is capital T small t. This is heterozygous allele. Understood? So here one is capital T, one is small t, that is one is dominant allele, one is recessive allele. So આમાં કોઈ એવી સ્ટોરી નથી કે જે તમને નથી ખબર. You know all the story, right? Okay, when it is into the heterozygous state, I repeat, when it is into the heterozygous state, જે એક્સપ્રેસ થાય એને કહેવાનું છે ડોમિનન્ટ, જે એક્સપ્રેસ નથી થવાનું it is called recessive. So this gives expression. Right? So only one out of these two allele will be expressed. જે એક્સપ્રેસ થાય છે that is called dominant, જે એક્સપ્રેસ નથી થતું that is called recessive. આમાં કોઈને કાંઈ ડાઉટ ખરો? Do you, any one of you have doubt or not? No. All right. That's fine. Okay. That's the first law of dominance. आज law of dominance છે. Characters are controlled by a discrete unit called factors, that is your alleles. Factors occurs in pair. There are two alleles. In a dissimilar pair, that is heterozygous, one member of the pair dominates, that is dominant, to the other which is recessive. એટલે એક dominant આવશે, એક recessive આવશે. જે એક્સપ્રેસ થાય છે એને તમે કહેશો dominant, જે એક્સપ્રેસ નથી થતું એને તમે કહેશો recessive. ડન છે? કોઈ ડાઉટ છે? ચલો, ઓલ રાઈટ. Rule number 2, second law. That also you know it. I તમને ખબર છે, બોલો. This law also you know it. Right? ક્યાં તમને ખબર છે? શું તમને ખબર છે? What do you know? What do you know? All right. This is parents, capital T, small t, small t. Okay. Gametes, capital T, small t. Why only one I am writing here, not two? Yes, Bhuj Center. Because say it again.
Homozygous parents produce same type of.
Okay, same type of gametes they are producing. બે શું કામ નથી એમ? કેપિટલ ટી બે ભલે ને સેમ હોય, મને શું વાંધો છે? બે જ શું કામ નથી? મારો કહેવાનો what I am asking is that question. Similar, dissimilar નહીં. Why there is not two and one? Yes, Ahmedabad Center.
Gametes तो two use करते हैं सर.
Say it again.
Gametes use करते हैं ना.
Okay, let me explain the logic. Right? Okay. Father, mother, both are 2n. Diploid. Okay. Child also is diploid. આમાં કોઈને વાંધો છે કંઈ નથી ને? ઓકે, હવે આમાંથી 2n આમાં ગયું, સે ફોર એક્ઝામ્પલ આમાંથી 2n આમાં ગયું, સે ફોર એક્ઝામ્પલ, તો અહીંયા થાય 4n. પ્લોઇડી ડબલ થાય, ભલે થાય કાંઈ વાંધો નહીં, ઇટ ઇઝ ગુડ, એવું માનીએ આપણે. એના પછીના જનરેશનમાં શું થાય? 8n. એના પછીના જનરેશનમાં 16 1. ઓહો, 1000ની જનરેશનમાં ખાલી ડીએનએ જ હોય, બીજો કોઈ મટીરિયલ જ ન હોય આખી બોડીમાં. સો ટુ અવોઇડ ધીસ સર્કમસ્ટાન્સીસ, આ 2n માંથી એક જ n અહીંયા જવો જોઈએ, 2n નો જવા જોઈએ, એક જ n જવો જોઈએ. અને I2n નો જવો જોઈએ, એક જ n જવો જોઈએ. સો અહીંયા શું કર્યું? ગેમીટ. અહીંયા શું કર્યું? ગેમીટ. Whatsoever gamete they have produced it by reductional division called meiosis. Chapter number 10 ની અંદર આ explain કરેલું છે. This is explained in chapter number 10, standard 11. That's why I asked this question, કે why not 2 and why 1? અહીંયા 2 છે, અહીંયા 1 થઈ ગયું. It means બે ભલે છે, બેમાંથી એકને જ જવાનું છે ગેમિટ્સની અંદર, એકને જ ટ્રાન્સફર થવાનું છે. Am I very correct with this now? So though they are living in pair into an individual, but when individual makes gamete, બે સેપરેટ થઈ અને ગેમેટમાં તો સિંગલ ફેક્ટર જ જાય, સિંગલ એલીલ જ જાય. This is its prime requirement. Clear? All right. That's what it is given here. એને કહેવાય લો ઓફ સેગ્રીગેશન. સેગ્રીગેટ થવું એટલે એગ્રીગેટ થવું એટલે ભેગું થવું, સેગ્રીગેટ થવું એટલે નોખું પડવું. સો વેન એક ગેમિટ બીજું ગેમિટ ફર્ટિલાઇઝેશન કરે ત્યારે એગ્રીગેટ થાય છે. કોણ? બે એલીલ. પણ જ્યારે એ જ ઇન્ડિવિજ્યુઅલ જે મોટું થયું, એડલ્ટ થયું અને ગેમેટોજિનેસિસ કરશે ત્યારે એ જ જે એગ્રીગેટ થયેલા હતા એ પાછા સેગ્રીગેટ થાય. So that is law of segregation. So during gamete formation, whensoever there is a gamete formation, the factor or alleles of characteristic pair present into parent will segregate to form, ready, gamete. Gamete formation માટે એ બે અલગ થશે. So during gamete formation, the factor of a character pair present into the parent segregate from each other such that a gamete receives only one of these two factor. એની પાસે બે ફેક્ટર હતા, બે એલીલ હતા, બેમાંથી એક જ ગેમિટ્સમાં જાય. Say for example, the example what they have shared here, કે if say for example it is homozygous dominant, so either this capital T will be going into gamete or this capital 2 will be going into other gamete. If it is homozygous recessive, either this small t is going into one gamete or this other will be going into the other gamete. If it is heterozygous, say for example, so one capital T, that is dominant allele, is going into one gamete and the other factor is going into the other gamete. So they are going to be separated while the gametogenesis is going to happen. જ્યારે gametogenesis થશે, they are supposed to segregate from each other. Fine. Any doubt? Any doubt? All right. Homozygous parent produces similar gametes. જો આવી રહ્યું, આ homozygous છે, આ homozygous છે. They produces similar gametes. But when there is heterozygous parent, they form two types of different gametes. એકની અંદર હશે dominant allele, એકની અંદર હશે recessive allele. So heterozygous parent produces two kind of gametes, each having one allele with equal proportion. Means, say for example, gamete 1000 baina. So 500 will be having dominant allele and 500 will be having recessive allele approximately. Ready? So equal proportion ma બે allele distribute થશે. So that's what law of segregation. Okay, any doubt into these both laws? All right. Now we are moving one step ahead, and that's inheritance of two genes. Now the second experiment of Mendel. So there are two characters which are getting forwarded. There are two characters which are getting forwarded from one generation to the other. Am I correct or not? I am not correct. I am not correct. જેટલા traits હોય, જેટલા character હોય, એ બધાય કેરેક્ટર એક જનરેશનમાંથી બીજી જનરેશનમાં એકી સાથે જતા હોય. Not one, not two, but all the traits are passing from one generation to the other. Now I am correct. Understood? Right? So now when monohybrid cross has been conducted by Mendel, right? He tried to find how many different traits it possesses, means pea plant has. But he knows, he knew, not knows, he knew that all these 7 characters are passing from one generation to the other simultaneously. એકી સાથે જાય છે. They are not individually passing. કે જાવે પહેલી બસમાં તું જા, બીજી બસમાં હું, પહેલી બસમાં હું આવું છું, એવું ન હોય. બધા હારે જ જાય છે. Understood now? So when they are getting forwarded from one generation to the other, do they have particular set of behavior or attachment with each other, or they are independently getting forwarded from one generation to the other? It means, simple formation માં સમજાવું તમને. 2 or 3 people what you are having right now in your class are studying same textbook, maybe same material, maybe into the same class. But when you pass your exams, it is not necessary into next standard, means next generation. હવે આપણે standard કહીએ. Next generation માં તમે જ બે same to same બાજુ બાજુમાં next bench માં બેસશો મેડિકલની સીટમાં. It is not possible. It may be possible, it may not be possible. Am I very correct? બેને હરખે હરખા માર્ક આવે, બાજુ બાજુમાં બેસી ગયા હો બોર્ડમાં કે નીટમાં, અને બેનું પેપર શેડ થઈ ગયું હોય તો વાંધો ના આવે, બેને હરખા માર્ક આવે તો એક જ સીટમાં બેસી શકો. Right? But generally It is not possible, right? Means first standard માં જે તમારી બાજુમાં બેઠું હતું એ અત્યારે નથી બેઠું ને? Simple. Am I correct? So when you are moving forward by one class to the other, maybe your benchmate, maybe your roommate, maybe your classmate changes. પણ બધાય જાય તો છે જ. Understood? હે ને? So dihybrid cross એને એટલા માટે ડિઝાઇન કર્યો કે he wanted to know કે when they are all together moving from one generation to the other, what kind of interrelationship they are showing, what kind of behavior they are having with each other while it is moving from one generation to the other. Understood now? The motto of this experiment is that. Okay, so this is the phenomena and the experiment is now dihybrid cross. Dihybrid cross. So there we are observing two different character, right? So character number 1, what he observed એનું first character હતું seed shape, right? And character number 2, seed color. There are two expressions of each character. બેયના expression બે છે. Dominant expression of seed shape is round. Recessive is wrinkled. Same way seed color, dominant is yellow and recessive is green. These are the traits. Now the factors of it. Round, wrinkled, this is R, this is small r. Yellow, capital Y, small y. So these are the alleles which are responsible to produce that particular type of trait. Okay, any doubt into this? અત્યાર સુધીમાં આ જે લખ્યું એમાં કોઈ ડાઉટ છે? Okay, now let us decide first phenotype of parent. This is the parent. Okay, homozygous, dominant and homozygous recessive. I need to take pair of alleles of each gene. Gene ની એ pair છે, allele ની એ pair છે, right? So gene are two. There are two genes. તો homozygous dominant માં first I am taking capital R any pair, capital R. Other I am taking capital Y and any pair. This is first gene, this is second gene. These are two allele of first gene, two allele of second gene. Doubt? Right? Okay. આમાં on the contrary આ dominant છે તો recessive માં એના small letter લખી નાખવાના. So small r, small r, small y, small y. So these are the genotype and phenotype. Genotype તો મેં લખ્યો પણ phenotype એ લખવો પડશે ને હવે. So here round, yellow, seeded plant. Round and yellow seeded plant. Here wrinkled and green seeded plant. So that's what genotype and phenotype. Genotype and phenotype of both the parents. So it is a cross between these two genetically different plant. So it is cross pollination between that. Okay. Gametes whatsoever they are getting produced from here are carrying each of these one. એટલે આની અંદર બે માંથી એક એલીલ અહીંયાથી આવશે કેપિટલ આર, એક એલીલ આનું આવશે કેપિટલ વાય. બેયનું એક એક એલીલ આવશે. Same way, it is also carrying two allele here, small r and small y. Right? Doubt? No doubt? Okay. So when it is the fertilization between these two, it will produce, say for example, capital R, capital Y, and small r, small y. So there is fertilization between these two to produce F1, carrying capital R from here, small r from here, capital Y from here, and small y from here. So this will be the genotype of all the progeny whatsoever in number generated into F1 will be having these genotype with these phenotype. Capital R, small r, તો phenotype શું થશે? Round or wrinkled? ભુજ? Round. Continue. Capital Y, small y? Yellow. So all, that is 100% progeny, will be showing these phenomena of genotype and phenotype. બેયમાં genotype અને phenotype બેય same હશે, જેટલી progeny હશે, right? Round and yellow. So it is almost exactly the pattern is followed of monohybrid cross. Yes or no? Right? ચાલો સેલ્ફિંગ કરીએ. કેપિટલ આર, સ્મોલ આર, કેપિટલ વાય, સ્મોલ વાય, આનું સેલ્ફિંગ કર્યું. રાઈટ? વી હેવ ટેકન the pollen grain of the same plant. We have give it to the other flower of the same plant, or we have taken the sibling plant and crossed it. So self pollination, I mean. Round and yellow, round and yellow. Right? Now comes the formation of gamete. હવે અહીંયા formation of gamete કરવું પડે. So you have one these and second these two genes you are having, and both have different types of allele. Both are heterozygous by state. So you have total 4 alleles. Am I correct or not? You have total 4 alleles. So you have two way to make different types of gamete. Right? એક છે તમારી પાસે normal method of Punnett square. Punnett square method. આ capital R બરાબર છે? Sorry. Capital R, small r, capital Y, small y. So that is the most common method what we can use it. So capital R, capital Y, first possibility. Small r, capital Y, other possibility. Capital R, small y, third possibility, and small r, small y, fourth possibility. So gamete number 1 gamete number 2, gamete number 3, gamete number 4. This is one method you can use to find out what types of gamete it can form. Okay, the other method I am using here. Look, R, R, Y, Y. So let me count first these R. It can form pair with these Y. So R, Y. Straight way I am putting. Or these one. These R can be making pair with small y. So capital R, small y. Other possibility. Now let me count these small r making pair with capital Y, or it can make pair with small y. So 1, 2, 3, 4, 1, 2, 3, they are same. Either way you can use it to find out what types of gamete they can form. So approximately both method are same. આમ તો બે મેથડ સેમ જ છે, બરાબર છે. આ ડાયરેક્ટ મેથડ છે, આ થોડુંક તમારે ઓલું ડિઝાઇન કરવું પડે ક્રોસ. All right, so total 4 types of gametes can be produced by both the parent. 4 from here, 4 from here. So you can have 16 progenies. 4 into 4, 16, right? So two generation, that is first generation તમે જોઈ, second generation F2 ની અંદર you have 2 parent. One parent, say for example there is a male parent, it has capital R, capital Y, one gamete, capital R, small y, the other gamete, small r, capital Y, the other gamete, and small r, small y, the fourth gamete. And you also have female parent which is having again capital R, capital Y, capital R, small y, small r, capital Y, and small r, small y, 4 types of gamete. So when I design Punnett square for these, say for example capital R, capital R, capital Y, capital Y, right? One more, I am doing it for you. Capital R, capital R, capital Y, small y. Okay, now you have pen, you have paper, and you have an exercise. Chalo, let you do that. આખું ટેબલ પૂરું કરો ફટાફટ. ચાલો, જીનોટાઈપનું જે સૂતા છે એ ઉઠી જાય અને ફટાફટ કામ કરવા માંડે. So here is the entire chart for you, includes genotype and phenotype both. Now it is purely has to made by you, right? કે ભાઈ કેપિટલ આર કેપિટલ વાય કેપિટલ વાય છે તો જીનોટાઈપ ઉપરથી ફિનોટાઈપ શું હોય, વોટ વુડ બી ધ ફિનોટાઈપ બેઝડ ઓન ધ જીનોટાઈપ વિચ ઇઝ યુ હેવ ગોટ ફ્રોમ that table, making that table, right? That you are supposed to design, that you are supposed to mention. It is very important for us, right? એ તમને આવડવું જોઈએ. Number 1, I have given you few examples. The same way you are supposed to follow. Say for example, here, so capital R, so it is round, and capital Y, it is yellow, right? Round and yellow. So that way you are supposed to design all the genotype and phenotype this way. So here capital R round, capital R small y yellow. So entire 16 different types of progeny with reference to genotype, right, and 4 different types of variety with reference to phenotype, you will get it. So phenotype you are getting R4, phenotype what you are getting 4. Okay, which 4 phenotype you will get? You will get round and yellow seeded plant, you will get wrinkled and green seeded plant. This you already had it into the parent. This used to be parent 1, this used to be in parent 2, this was never found in any of the parent. So when I count the number of round and yellow individual, 1, 2, 3, 4, 5, 6, 7, 8 and 9 individual I will find. Pattern would be this only, pattern. Look at the pattern now. This pattern. Round yellow, 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 round yellow. A pattern fixed છે. Ready? Okay, second pattern જોઈ લ્યો અહીંયા. ચાલો. Round green, આમ. Round green, આમ. જુઓ જો. મળી ગઈ? Okay. Wrinkle yellow, જોઈ રહી. Same pattern, આમ. હવે તો એક જ વધ્યું છેલ્લું, wrinkle green. So total 9, પછી 1, 2, 3, 1, 2, 3, 9:3:3:1. That's the phenotype ratio. So round and yellow you will find 9, round green you will find 3, wrinkled yellow you will find 3, wrinkled green you will find 1. So total 9 plus 1, 10 out of 16 will be parental combination. And 6, 1 and 2, 6 out of 16 will be recombination. Ready? That is called recombination. So let us conclude today's lecture. આપણે અહીંયા આજે અટકીએ છીએ. Later discussion will be made tomorrow or tomorrow onwards, right? So we have finished dihybrid cross. We are into the conclusion of dihybrid cross later on into the next lecture. Fine. I'll see you. Take care.